The last speaker of this session is Professor Dr. Sarah Michel, or Michael? Michel. And she's from University of Maryland, Baltimore. She's going to speak about evaluation of iron species in subjects treated with generic and reference ferric gluconate.
Well, thank you very much. Yeah, I'm really the person before you on lunch, so hopefully you'll bear with me and listen to a story that I have about a clinical trial that we're going to start soon at Maryland. And I also want to thank Dept and CLINAM for the opportunity to come here and speak. This is a project that we're working on in collaboration with the FDA at Maryland. My co-PIs are Maureen Kane and Jim Pauly, and at the FDA, Daejun Sun, Nan Zhang, and Wenli Jiang. And so we're looking at sodium ferric gluconate. In the US, there are 6 iron drugs that are approved, of which there's one that there's a generic counterpart, and that's the one for sodium ferric gluconate. Ferric is the name of the brand. It's an iron oxide complex. It's a nanoparticle complex. It's made by taking the iron oxide, adding the gluconate ligand, and then you get this complex that's stabilized by sucrose. And so the points of giving iron, iron is given intravenously to patients who have severe anemia. Often these are people that have chronic kidney disease. And the idea is the iron is given by IV, it's processed through the RAS, and it gets to the plasma where it binds to a protein called transferrin. Transferrin then brings the iron into the cell, and it will fill up the proteins that are deplete in iron due to the anemia. If, however, too much iron is delivered, the transferrin will become saturated. Iron can no longer get into the cells through the normal pathway, and you have a portion of the iron that's collectively called non-transferrin bound iron. And so this is iron that can be bound to other proteins in the plasma. It's typically albumin or ferritin, and also iron can be bound to small molecule chelators, principally citrates. And this pool of iron, called non-transferrin bound iron, or NTBI, can also get into the cell. It goes through these non-specific transporters. Once in the cell, this is called the labile iron pool, and it can cause some toxicity. It can react with oxygen to form reactive oxygen species that can then damage proteins, DNA, and lipids. And so the question that we're asking in our trial is, do the brand and generic sodium ferric gluconate deliver the same amount of iron? And the impetus for this was based on some studies in Europe where the brand and generic, when the patients were switched from the brand and to the generic, and there was some toxicity observed. And so we're going to do a clinical trial with 52 healthy subjects. They'll be given either the generic or the brand drug, and then we will measure the iron speciation, and then they will be brought back in again and be given the other drug. So it's a crossover study. It's a PK study, so we're going to collect a lot of blood samples. So in red, this is where the red arrows indicate all the iron measures we're going to take. We're also going to take some secondary measures, oxidative stress, lipid peroxidation, and some safety measures. Let's see. So what are we looking for? So in the plasma, we expect to see some different iron species, and this slide is showing you what we expect to see. So in addition to the drug shown here and transferrin, we also expect to see iron associated with albumin, iron associated with ferritin, and then principally iron citrate. And so these are the species that we're going to be looking at as our iron species in our clinical trial samples. And so I want to give you some terms that we're going to be defining as I go through the bioanalytical assay that we're developing. So we're going to measure total iron, so that's all of the iron in the plasma. Then we're going to measure transferrin-bound iron, that's TBI is the acronym here. We're going to measure drug-bound iron, then what we're calling protein-bound iron, so it's iron associated with these proteins other than transferrin, and we're going to measure labile iron. And protein-bound iron and labile iron collectively is the non-transferrin-bound iron. And so the question that we first had to address before we started this clinical trial is how are we going to measure all of these different iron species? So this, if we look at this side, these are where all the different species are. We're going to measure the transferrin-bound iron. We want to measure the iron bound to these other proteins and the labile iron. And we also when we started working on the analytical assays, we also were hopeful that we might be able to also measure the drug-bound iron. This had not been seen before, measured before. And so the strategy that we came up with to do the trial is we're using inductively coupled plasma mass spectrometry as our main readout. So ICP mass spec is an assay that allows you to quantitate metal ions in solution. So you can look at small molecules, you can look at iron associated with proteins, you can go all the way up to tissues. It's also very sensitive. You can typically go down, in principle, to parts per trillion. And so our strategy was we'll have our plasma samples, we'll measure total iron directly by ICP mass spec, because here's just one iron measure. And then then our idea was that we could hook the ICP mass spec up to an HPLC and separate out all of the other species, and so measure all of the different, the iron distribution. So for total iron, this was the easier method to validate. We did method validation by taking our plasma and we doped it with a series of different concentrations of our drug to mimic the amount of iron drug that the patient would see when you give them the IV. And so we did a series of experiments and we were able to method validate total iron. And I just want to show you sort of our data. So we followed the FDA guidelines and it works great for the whole concentration range at which we expect to see iron. And as a comparison, so in this slide, the red is our ICP mass spec data. The gray is the expected amount of iron. And the blue is another assay we did for comparison. So the other assay we used ferrozine, which is a more traditional way of measuring total iron. It's a colorimetric assay. You reduce the iron to iron 2 and add ferrozine, and you see a change in the UV vis. And what we noted was that our assay for ICP mass spec actually is a little bit better. We're able to see more accuracy over a larger range. So we have this assay all set to do for our clinical trial. The other assay, though, is how are we going to look at all the different species in our plasma? How are we going to look at the transferrin-bound iron versus the labile iron and so forth? Now, traditionally, there's a lot of people have thought about this, and there are a lot of different ways that people have measured what we call collectively NTBI. And often what people do is they use a chelator, so NTA, or here are some different structures that people use. You chelate the labile iron or the non-transferrin-bound iron, you see some kind of change, and from there you're able to measure the non-transferrin-bound iron. A drawback to this approach is whenever you put a chelator into your solution with plasma, you potentially can perturb the equilibrium because what you're seeing is iron bound to all these different species in the plasma. And there was a hint that perhaps ICP mass spec could be used if it was hooked up to an LC. And so we decided to pursue that as our assay to measure all of the different iron species. And so our reasoning was we weren't going to add a chelator, we'd be able to just look under X condition at all the different species. And so here's our setup. We have a bio-inert HPLC, so that means it's all metal-free. We're not introducing any new metal. And we've hooked it up to our ICP mass spec. And so you can take your plasma, put it over a column. You can measure the different proteins or iron species just through the, looking at the UV with a HPLC. But then once you're hooked up to the ICP, you can actually measure the total iron content with each peak. And so the y-axis here is iron content. And so in thinking about this, we had to sort of think about what was the right range to do, would it be suitable? And so we knew approximately how much iron is in the plasma to begin with, so these are published numbers. We also had an idea of how much iron we would see before and after infusion with the drug. And we also knew the molecular masses. And so our strategy is to use size exclusion chromatography, so we needed to be sure that these masses were fairly separate so we'd be able to separate. And so this is work done by a really talented postdoctoral fellow in my lab, Heather New, and she spent a long time trying a lot of different columns, and what she found was that if she put 2 columns in series, these bio-inert size exclusion columns, she was able to get really nice separation of the different species. So she can get the drug, she can get, we're calling protein-bound iron, so that's iron bound to either the ferritin or the albumin. She can get transferrin-bound iron, and then she can get labile iron. So we're able to simultaneously measure the iron content in our plasma associated with all of the different binding partners that are present. And so this is just to show you where we are. So we're going to start the clinical trial quite soon. We've validated total iron. We're almost finished method validating all of the other measures. We still have some of the longer-term storage samples to get the data for, but we're going to start this clinical trial in July 2017 and use this method to measure the iron speciation. We also have to do some secondary measures I mentioned. So the secondary measures are oxidative stress and some other some inflammation markers. And what we're doing here is we're principally using a mass spectrometry approach as well. We have a fabulous facility at Maryland, and so working with Maureen Kane, there are several different things we'd like to look at. So if there's excess labile iron, you can generate reactive oxygen species. If we see increased oxidative stress, we're going to look for these two markers here, heme oxygenase, and then we have a nice mass spec assay for glutathione disulfide. Increased inflammation, we're going to use a cytokine panel and a mass spec assay for C-reactive protein. Lipid peroxidation, we have a melanodialdehyde mass spec assay, and then we have a citrulline assay for reduced NO synthase. And so finally, I need to thank everyone that's worked on it. We call ourselves Team Iron. Heather is this fabulous postdoctoral fellow in my lab who did all the LC ICP mass spec. Joel is working on the physiochemical characterization of the iron drugs, which I don't have time to show you today, but I learned a lot of good things yesterday when I heard folks talk about those drugs. Annie is a pharmacy student that's been working with Heather. Maureen is our mass spectrometry colleague who's doing the secondary assays. And then Jim Pauly, some of you know, is a terrific collaborator on this who's really organizing the clinical trial. And then I must thank the FDA who's supporting this project. And thank you for your attention before lunch.
Okay, so I'm sure there's a lot of NBCD folks here who would have a field day. No? Questions?
Any questions?
I have a question. I presume you're looking at, I mean, the ultimate goal of this is to evaluate safety of different
That's right. It's a safety
formulations,
study.
Of course. And you're using sort of the PK and this detailed analysis as a surrogate.
Yes.
For safety.
Yes. Yes.
Because in a 50-patient
That's right. That's right. We're limited by. That's right.
Yeah, you
yes.
can't evaluate safety. Given the different types of iron, you know, there's gluconate, sucrose, there's a few others
right.
which I'm not very familiar with. How do you sort of think of sort of the endpoint of this when you get the results of the study? Will you have a conclusion? I mean, in terms
Right.
of the ultimate goal, which is safety.
Right. So this study is just on the gluconate, and I think we'll be able to potentially suggest, you know, if there were, you know, differences seen in patient populations, it's because, you know, you're seeing the iron distribution change, right? I think we're going to get a nice picture of what's going on with regards to the iron distribution. The other thing I think we're going to learn from this is we're able to actually track the drug, right? Because we can separate the drug as well, so we can measure how much iron is associated with the drug. I think this would be terrific then to use for some of the other iron preparations. You know, I guess I don't know sort of if other generics are sort of in the pipeline for the US, but my guess would be potentially, and I think that would be a nice way to sort of get sort of data that you can compare for all of the different drugs. I think one of the things that sort of, there's a lot of, I wouldn't say discrepancies, but people have used different techniques to measure non-transferrin bound iron, and so then how you compare the data I think is a little bit difficult, because if you're using a fluorescent probe, you may see some data that's you know, pulling too much metal or not enough metal. So I think here we're really solving this problem of how do you measure the different species.
Question here. 30 seconds, remember.
Yes, a quick question. It's not the same to give your product to a person that has anemia than to give iron oxide nanoparticles for many of the applications that we have to a patient with maybe a cancer or autoimmune disease or whatever. What is the range? What is the difference if you have a healthy individual and you treat with an iron spion?
Oh.
So it's very far from the doses that they are giving and they are then present in the healthy individuals?
So that's a great question, and I'll try to answer the best as I can, because I'm a biochemist by training, and this is sort of a new area for us. So you're right. So I think when you're treating patients who have anemia, right, they have lower iron, and so this is, you know, from what we understand, giving healthy volunteers this drug, it will also, you know, we don't anticipate it having sort of terrible toxic effects. But I guess could you sort of clarify your question a little? I'm sorry.
and it will be different in a healthy individual or in a person that has anemia. And maybe you will give a certain dose, and the amounts that you will, the total iron in the body is not with all, it will be a lot lower in an anemic person, even with the treatment, that in a person, maybe it's not healthy, it's healthy because so you treat with a nanoparticle made of iron oxide to treat their disease, but the iron is not the problem of this person, so you will have an overload of iron. Is this a risk?
Oh, I don't think we anticipate that there's a risk of an overload of iron. I do think, though, a follow-up study would be to look at patients with anemia, right? Because then you'd really be able to work out what's going on. But we don't anticipate there to be a risk.
Any other questions? Last question. Beat is telling me to wrap up, but to be fair to the speaker, she finished on time. Again, sort of the end game. I mean, you're developing this detailed characterization method, which is very nice. But when you, in the end, compare, and let's say you end up with the same results for, say, a generic versus an innovator composition, what is your conclusion? I mean, you still haven't answered the safety question, right, at that point? Or, I mean, maybe you know a little bit more. But how do you, because the risk of, as I understand, there is risk of anaphylactoid, there's all kinds of other risks, and there's deaths reported, I believe. I think that if I remember right, it was about 20 per 100,000 or something like that. Might be a little wrong, but how do you end up with a conclusion on safety at the end of the study?
Well, I think we're going to learn sort of fundamentally sort of a little bit more about how this drug works, right? And how I think we're going to be able to link sort of the properties of the nanoparticle and differences we see in the nanoparticle properties to sort of what's fundamentally happening on the biochemical level. I think that to further study safety, we'd probably have to do some additional follow-up studies, right? So I think we view this sort of as a first step. We have this assay that's now going to let us sort of compare these two different drugs and understand if sort of what's going on in the cell, the biochemistry, is the same. So I think that's sort of as far as I can tell you about that.
All right, thank you very much. And let's thank all the speakers in the session for a very nice conversation.